Paul will replace you. Come up, Paul. Come up, Paul.
So, thank you, Dusan, for the nice presentation. I'm Cristina, student from LandLab, and I'm the scientific director in LandLab. So I followed the P4P project since the very beginning. Not since the very beginning, because the very beginning was in 2000. I was in high school, but since the very beginning of my job in LandLab. I heard the screen sound. Okay, I go on. No, it's still there, the sound. Okay, so it's still there, the sound. I can also do it now, maybe.
Yeah,
it's a small room. Okay, so yeah, you managed. Thank you. So yes, this presentation was very hyped up yesterday, very promoted. So if you're disappointed at the end, you go there, and what is there, and complain with them. Okay. And anyway, if at the end you still have some questions about everything, you can ask me, and it will be, I think, answers coming from different parts of the room. Okay. So. Sorry. Yes. So now what I want to present today is what we know about the mode of action of the P4P products that we have now in the market, which are Forterra, Forvita, and For Good. Okay, let's start with Forterra. Forterra has been developed to increase the NOE in crops, and it can be applied both in plants, both in comfort and sub-comfort conditions. It is for fertigation, and since yesterday there were quite some questions about when it should be applied better, we recommend that it is applied through different fertigations, at least 3 applications, even more if possible, and at the beginning of the vegetative growth, at the beginning of the crop vegetative growth, because it's when phosphorus is more needed for the plants. Okay, now the right dosage I cannot remember. I think it's 16 liters per hectare divided in split applications. So actually we wrote here, should I point there, or you can also see? No, you can also. Okay. So we wrote here NUE, so nutrient use efficiency in general, but actually Forterra was developed first to improve phosphorus use efficiency. Why? Well, because phosphorus, you all know that it's one of the main micronutrients for the plants. It is responsible for a lot of very important pathways and very important metabolism inside the plant. And the deficiency in phosphorus are shown as stunted growth, and the leaves turn purple and dark, and photosynthesis is hindered. So, of course, it's very important. From the point of view of plant nutrition, phosphorus in the soil can be divided in different pools of different availability, okay? So the majority of the phosphorus is actually not available for the plants. Only a minute fraction of that is available. And so that's why we need fertilizers. In fact, fertilizers are indeed help the feeding billions of people since 60, 70 years. Even more maybe, but phosphorus derived from phosphate rock, which is a no renewable resource that is concentrated only in few countries, in particular Morocco. And moreover, the inefficient use of phosphorus is polluting our rivers and our oceans, causing eutrophication. So it's a big problem. And in fact, in Europe, we have a lot of regulation for that, or there are starting to be a lot of regulation for that. So what we can do to help increasing phosphorus efficiency with our plants, there are different methods. There are some management practices. There is breeding of plants that are more efficient in using phosphate. And then there are biostimulants, okay? And then here is, that's why we are here now. So there are different biostimulants in the market that are known to be able to improve phosphorus efficiency to a certain level. Humic substances, seaweed extracts, phosphorus solubilizing bacteria, and mycorrhiza. Forterra, of course, you know, it's a plant extract, so it's something different, completely different than these four main categories. And it has also a very different mode of action compared to these four. So yesterday you saw some demo trial, very nice trials that were performed also thanks to you last year and the year before, but actually we have a lot of scientific trials where we analyzed much more in detail what Forterra was doing. And so I will show you some of these trials. In this case, we treated strawberry with Forterra, 3 times UC, so split application more than once in fertigation. And what we saw was there was indeed an increase. Let's see, okay, anyway, an increase in yield, in particular at the first pickings, okay? Yes, on the first pickings. We were worried about you. I was.
How did I make him up? With a lot of force.
Sorry for bothering you.
No. Well, we're starting now with the most important part, so don't worry, you didn't lose too much. Yes, so an increasing yield, in particular at the early beginning, at the first pickings, which is very good because, you know, it's when you earn more. If you have first the product, you earn more. So at the end, we got a 20% increase in yield. We didn't have a lot of differences in quality, but the fruits of the best size were more represented in plants treated with Forterra. So we applied Forterra in comfort conditions, at least in the so-called comfort conditions. Okay, so everything was perfect. It was farmer practice. Everything was, in theory, perfect. Okay, I said that here we are increasing phosphorus use efficiency. So why I'm saying that if we applied Forterra in comfort conditions? Well, because then we analyzed the phosphorus level in the leaves of the plants, and we didn't see any difference. So I'm still not answering the question. However, when we analyzed the level of phosphorus, available phosphorus in the soil at the end of the trial, we saw that there was an increase in available P in the treated plants, in both at the low dosage and the high dosage, but much more, of course, in the high dosage. Am I pressing something wrong? Okay. In particular, at the high dosage. So we had the 13% more available phosphorus in treated plants at the highest dosage compared to the untreated one. This is another trial where we applied Forterra in phosphorus deficient conditions because the plants had 30% or less phosphorus compared to the untreated plants. We saw a very nice effect on coloration. These were gala apples. So it was, coloration is very important. So it was nice to see that there was such an increase. What was not that nice is that we didn't see differences both in yield and in these quality parameters between the untreated. And from the scientific point of view, it's not very nice, okay? To don't see any difference between the negative and the positive control. But then again, when we analyzed the phosphorus level in the soil, in the leaves and in the soil, we saw that, first of all, there are differences between the two controls, with the full phosphorus having more phosphorus in the leaves compared to the decreased phosphorus. And then what's more important, we saw that with the addition of it's written LL4 because it was this first name, then it's Forterra, of course. With the addition of Forterra, we reached the same level of phosphorus as the control at the same, with the highest, higher pill level. And in the soil, yeah, well, we saw exactly the same thing that we saw with strawberries. So there was an increase in available phosphorus for both the dosages, with much more effect when the dosage was higher. The higher dosage here is exactly what is recommended now, okay? Because this is what is recommended now, and this is actually less than half. So, hello. Oh, don't worry, of course.
Rudy was not the last one.
Thank you, Dushan, for the presentation.
It was really nice.
Anyway, so you see exactly now. I will go on now. We see that there is an increase in available phosphorus in the soil with the treatment with Forterra. Very nice. But now the question, so yes, we need less fertilizer. But now the question is why? Why do we have such an effect? Is it a direct effect on the plants, or it is an indirect effect on the plants through the microbiome? Okay, so what we did to see what was happening, and in particular, if we could have a direct effect on the plant, we treated sugar beet seedlings grown in sterile perlite. So you can imagine there was no microbiome, okay? Or it was very, very, very limited. And we treated it twice. We kept the sugar beets at very low P levels, so 33% compared to the positive control. And we treated twice with Forterra. After that, after 3 days from the second, 3 days from the second application, we did the assessments. And here you see, first of all, that even just after 10 days from the first application, we have a partial recovery of the phenotype, because we got the plus 35% shoot compared to the negative control, okay? And what is even more interesting is that we saw an increase in the expression of the chloroplast orthophosphate transporter PHT2.1. What does it do? It has a dual function. When there is enough P, okay, enough phosphorus, that's translocating, uptaking the phosphorus in the chloroplast. And you know, chloroplast is where photosynthesis is happening, is where the life of the plants begins, okay? And when it's when phosphorus is very scarce, what does it do? It's retranslocating the phosphorus from the older leaves to the younger ones, okay? So it has plants keep on growing. It doesn't stop the plants doesn't die, doesn't die, because the phosphorus goes where the most photosynthetically active parts of the plants are, okay? So indeed, from this trial, we proved that we get a better phosphorus utilization, and in this case, Forterra application had a direct effect on the plants. It could not be mediated by other things. With this other experiment, we treated tomato plants with Forterra, both in comfort and subcomfort conditions, slightly subcomfort conditions, because we got 30% less phosphorus. And I can't remember who asked this question yesterday about about the sandy soil. Yes, exactly. And this was done in sandy soil, very sandy soil, 70% sand and 30% soil. This is an artificial substrate, okay, because we use it for different reasons, but it is sandy soil. And we analyzed the mycorrhiza colonization after 60 days from the second treatment. What we see here, well, first of all, there is an effect on yield, so more than 20% more yield. And then we had a very nice effect on mycorrhizal colonization. Here you see these are roots where there is some mycorrhiza, yes, and these are with much less mycorrhizal colonization. And here you see that in P subcomfort, okay, we got 70% more colonization compared to plants with the same level of P. And okay, there was also a slight increase when plants were in comfort, although it's not statistically significant, you see. So as you know, mycorrhiza are not just there just to help plants uptake P, but also all the other resources, all the other nutrients, even water. So from this, we can start to see why actually Forterra is not just there to increase, it's not just helping plants with phosphorus, but most probably also with other nutrients. And even more now with this trial that we performed in lettuce, and in this case we analyzed the microbiome of the rhizosphere 10 days after the second application. And first of all, okay, again, effect on yield, both at sampling time where we had plus 21% yield, and at harvest where we had plus 14% yield. This trial was performed in comfort by a foreigner, so it's something completely in normal conditions. But then when we analyzed the rhizosphere, we saw that there was a dramatic increase in the microbial population. You see here, much more DNA per gram of soil, and of course much more 16S gene copies. Okay, so it's 50% more DNA inside the soil, which is quite a lot, quite a booster of the microbial population. But now there are good bacteria and bad bacteria. So who did we influence? Okay, so I repeat this, but to make phosphorus, organic phosphorus, available for the plant, there are different enzymes, phytases and phosphatases. So what we did was to analyze the amount of bacteria that could produce phytases and the activity of the phosphatases of the soil. And here you see that there was an increase in bacteria that could produce phytases, so they could help phosphorus being made available for the plants, and which is very nice. But okay, we didn't see anything about phosphatases. It's happening anyway. A good effect on phytases for sure is making phosphorus more available. And then what is even more nice is that we got an effect also on the bacterial population that is involved in nitrogen fixation. Where is nitrogen fixation? This one. Okay, see, 6.6%, and also in nitrogen fixation. Nitrification. See, the difference is quite big. So we are not just influencing only phosphorus-related reactions, but we are also influencing other things, other cycles. And the same thing we see when we analyze the activity of some other enzymes involved in other cycles. Because apart from the hydrolysis of ketone, we see an effect on enzymes involved in nitrogen cycle, in cellulose cycle, in carbon cycle, and in sulfur cycle. So indeed, what we see in all the trials cannot just be explained by an increased PUE, but it must be due also to other factors, also to an effect on other nutrients. And this is something that Laurent actually was saying since 5 years ago. It cannot be just PUE. It must be also something else. So to conclude, Forterra has a direct effect on the plant, both a direct effect on the plant with better phosphorus uptake and utilization, and it has also an effect on microbiome. And I hope that I showed you that this is not just PUE, but also NUE in general.
If you look on the results on the apples, trial on the apples, you saw increased firmness. Firmness doesn't come from phosphates.
That's also true.
Yeah.
The increase was not very, very much, but it was statistically significant. Yes, indeed, the calcium and So indeed, that's also another good thing. So now let's go on with Forvita. At the end, you can ask me the questions. Now we go on with Forvita. Marine? Forvita? Okay. No, yeah, I talk. Okay, so Forvita has been developed to increase water use efficiency in plants. Okay, in all kind of plants, so all kind of crops, both in comfort and in stress conditions, and it should be applied again in split application 2-3 times with 10-14 days interval. And okay, since I will explain later what is its role exactly, but believe me now that when I say that since it has this kind of role also in comfort condition, it can be applied almost every time. Okay, its benefits before the start of a stress that you can forecast, but even if it's applied when there is no forecasted stress, it will be almost for sure do something. Okay, a lot of trials that you saw yesterday, they were in comfort conditions. Now, what kind of stress you are talking about? Yes, we say water use efficiency, it's increasing water use efficiency, okay. But from our trials, we saw that actually Forvita is doing something against oxidative stress. What is oxidative stress? Forvita, yes, also for good has this kind, but I will say that at the end. Yeah. Yeah, you can smell it if you want. It's not toxic. Whatever. Yeah, it is also nice. And sorry, what is oxidative stress? Oxidative stress is a natural occurring phenomena that is happening in all living organisms, also us unfortunately, since it's the natural production of a lot of different metabolisms inside the cell. However, there are some different environmental conditions that we call stress that can dramatically increase the amount of oxidation, the amount of oxidative stress that is represented as reactive oxygen species. Usually there should be a balance, okay, between reactive oxygen species production and scavenging, which is so the detoxification of the reactive oxygen species. But sometimes, in particular, if there are stresses, this balance is broken, okay? So we got much more reactive oxygen species, and what they do is they degrade, they break down a lot of different molecules like lipids, proteins, DNA. So they do quite some damage, okay? That's the reason why also we get older, and because there is also production of reactive oxygen species. So, of course, you can imagine that if you start degrading such an important molecules, the plant is not faring very well. In fact, we had a decrease in biomass, in yield, in quality, and to some extremes, we can get also plant death. So now let's see how Forvita can do something about this oxidative stress. I will show you some results. You saw a lot of results yesterday, but I will show you some more with some more scientific in-depth explanation. In this case, we treated the sugar beets. Again, I don't know why we treated sugar beets. Anyway, twice with 7 days interval, and then we applied a very, very strong stress. Like we didn't irrigate it anymore. And when they were going to die, almost die, we stopped it. We recovered them for 14 days. Okay, so we gave them back water and we recovered them for 14 days. What we see here, there are a lot of things, but you see that at the end of the recovery, the plants treated with the highest dosage of Forvita, then again, it's the one that is recommended, we get a partial recovery of the phenotype. Okay, there are no statistically significant differences between the plants that were kept in well water conditions all the time and the ones that were in drought stress and treated with Forvita, at least for the roots. There are differences for the shoot, okay? But still, there is a partial recovery, okay? And then we analyzed the amount of reactive oxygen species inside the plants in two different time points. In light color, yeah, here in light color. In light color, there is the sampling at 3 days after the start of the stress. Okay, so at the early beginning, when the dark column, it's at the end of the drought stress. So when we decided to recover, and then you see here the treatments with the Forvita. You have good eyes, I think you can see also this one, right? So treatment with Forvita were indeed decreasing the amount of hydrogen peroxide, which is a reactive oxygen species, okay? And this was particularly true with the highest dosage, which is the one, again, recommended. What does it mean? It means that either there is less reactive oxygen species production to begin with, or there is higher antioxidant activity, because if so, that there is the balance, and it can be both. It can be both ways. So we looked at the activity of some of the antioxidant enzymes that are involved in detoxification from reactive oxygen species, and this is catalase and glycol peroxidase, and we saw that actually the activity, it was not at all increased. Look at this. This is exactly as the control, and this is even lower than the control. What does it mean that? It means plants are not stupid. If they had problems, they would try to solve them. Since they didn't activate these enzymes, it means that there were actually less reactive oxygen species production. So with the next trial, I hope I will explain a little bit how this could be. So we performed another trial, which was copy paste more or less of the other trial, but on tomato. And And in this case, you see, during the drought stress, we assessed by visual scoring the wilting of the plants. So there were untreated drought stress plants, which is the negative control, for Vita drought stress plants, which is the treated plants, and the UTC well water, which is the positive control. Of course, 5 is the best score, so it's perfect plants in perfect conditions, while 1 is a dead plant. Yes, here you see that it started to show something 4 days after the last treatment, so the drought stress started to kick in. And at the beginning, you see that the plants, both negative control and treated plants, were going more or less equally. But then you see, when we decided to recover them, you see that the plants that were treated with Fovita stopped wilting. Okay, they stopped the process of wilting, which means that we're able to put in place mechanisms to cope with the drought stress, with the lack of water. We also analyzed the photosynthetic efficiency. Now, I'm not going to explain all of these strange symbols, and I always forget what is phi and what is psi. There are Greek guys, so probably they can help. But just to show you that at 4 days after the last treatment, so when actually we didn't see any difference, we didn't see any visual difference between the plants, the treated and the untreated plants. There was no difference. However, we saw that treated plants started to have higher photosynthetic efficiency. They started to have less dissipation of the excess energy through heat, which is something still that the plant doesn't like. And in general, all the photosystems, also the dark reactions, this is the performance index, were much better in the treated plants. Okay, so in fact, above us, so see, the higher the better, and the highest is this one. The lower the better, and the lowest is this one, the treated plants, and the higher the better, and the highest was the treated plants. Okay, so you see that actually all the photosynthesis was improved with the treatment of Forvita, even when we still weren't seeing any changes in the plant's phenotype, or very, very small changes in the plant's phenotype. And at the end, you see that we got an increased biomass. So after 14 days of recovery, we get an increase in biomass compared to the negative control. We still couldn't reach the well water because it was really a strong stress. I mean, the plants were really kaput. And we also saw a slight increase, in number of flowers and fruits. So actually, this is not statistically significant different than this one. Still a little less, meaning we could not recover everything. But it's a much better behavior. Now come a little bit the tricky part. And because to investigate why this was happening, sorry, Marin, we had to do the metabolomic analysis. And what is metabolomic is the study of the metabolome, which is the complete set of metabolites in an organism, in this case in a plant, in a certain moment. Okay, and this is the result of all the things that happens here. So from DNA to RNA to proteins, then there is the metabolome, so all the molecules that are actually making what we see. They are making the phenotype. Okay. So it's the step right before what we can see with our own eyes. And we analyzed it, and now also this is a little difficult. So these are the metabolites that are present in different quantities compared to the well-watered plants, okay? And you can see here the untreated plants in drought stress conditions modulated the genes that are in the colder colors, let's say, while the plants treated with Forvita modulated this amount of genes. Okay, some of them are in common between the untreated plants in drought stress condition, but a lot of them are not. Look at here. So you see that Forvita is modulating much more, is making plants modulating much more metabolites. And most probably, these metabolites are the ones that are helping the plants to cope with the stress that they get. Now let's see. We said before that reactive oxygen species, one of the reactive oxygen species bad effect is the oxidation lipids. So the lipids are on the membrane, so they are pretty important. All organelles on cells, so they are pretty important. And what we saw is really a remodulation of all the lipids of the cell. Here you see that the negative control doesn't change almost anything compared to the well-watered plants. So see 0, no change, 0, no change, 2. Yes, the PG, which are the phosphatidyl glycerols, are a little bit more expressed, but not that much. Look at what 4-beta does. Okay, it's decreasing a lot MGDG and VGDG and increasing much more PG, phosphatidyl glycerol. And we know from literature that phosphatidyl glycerols are very important to preserve the photosystems inside the chloroplast. Okay, we know that from literature because there are studies with knockout plants. So this is known. Okay, so 4-beta is preserving the plants from drought stress by remodulating all the lipidomes, so all the lipids of the membranes, and this helps preserving the photosystems inside the chloroplast. And actually, if you remember two slides ago, we said that there was a better photosynthesis also. So this is really completely in agreement with what we saw with the physiological analysis. And then now there is a little bit difficult part about photosynthesis and about chlorophylls. So it would be nice if photosynthesis could just be described like that. This is for you, Marin. And so just plants take water and CO2 and light and transform them in oxygen and sugars that are needed for their own growth. But actually, it's more like that. Okay, it's a little bit less fancy, a little bit, and it's divided in light dependent and light independent reaction. And what's even worse, look at this image, Marin. This is what happens in the light reaction. Now we're not going to go into details, just to show you where we should focus now to explain what is happening, which is the very first step of the photosynthesis or of the photochemistry, which is the harvest of the light. The harvest of the light is made by chlorophylls, of course, which are the pigments in the photosystems. And what is happening when a chlorophyll molecule is hit by light? So harvest light, we say harvest light. It gets excited. So it goes to an excited state. And from there, it has to go to the basal state through different mechanisms. So we have photochemistry, which is what the plants want, what we want, because with photochemistry, it can produce all the glucose and so on, and the oxygen for us. That is, you see here, there are, okay, I will explain it later. Anyway, photochemistry, then there is fluorescence. Okay, that's not important right now. There is dissipation of energy through heat, of excess energy through heat. So this is a protection mechanism for the plant. And then there is this other pathway that is always present whenever there is light hitting our plants. This pathway is always present, although the best thing would be if it was reduced, because what this pathway is doing is producing reactive oxygen species. Okay, this is something that is always happening when the plant is doing photosynthesis. The important part is that the left. Oh, I have a problem with that right part is more, let's say, the energy goes more in the right part of the figure, not in the left part. Now let's see what happens by analyzing the metabolites in the treated plants. Okay, just here to show, this is a very simple way to show the beginning, like all the photochemistry. So we get the light harvesting complexes that, as the name say, harvest light, okay? And they are formed by chlorophyll of two types, A and B. While there is the core system, where there is the reaction center that really start all the process that is made by chlorophyll A dot, okay? What we saw with the treatment is that the untreated plants decreased the quantity, the levels of just chlorophyll A, while in for Vita treated plants there is a temporary decrease of both chlorophyll A and B. Okay, what does it mean? It means that in UTC, the untreated plants, the loss of chlorophyll A probably means the loss of just the reaction center, because you cannot lose these ones without losing also chlorophyll B. While with the 4-beta treated plants, we lose both chlorophyll A and chlorophyll B, And so it means, and actually by calculating the ratio is the same as the light harvesting complexes. So it means that there is a remodulation of the light harvesting complexes. You think it's a bad thing? It's not. This is exactly what plants do when they have to deal with this kind of stresses. They remodulate the light harvesting complexes to be able to survive better with what is now becoming highlight, because everything else is getting blocked a little bit. So with the light that was perfect in stress, it becomes too much. There is a saying, too much of a good thing can be a bad thing. In this case, it's like that. And so this disruption of the core in the untreated plants is decreasing the photochemistry, increasing probably all other mechanisms, but it's also increasing reactive oxygen species production, because the energy has to go somewhere. So if it doesn't go through pathway 2, it has to go through 1, 3, and 4. Okay, so there is higher reactive oxygen species production. While in 4-vita, with this rearrangement of the light harvesting complexes, we get lower light absorbed, but it's also less stress, an optimization of all the reactions, all the photochemistry, which we saw also in the photosynthetic parameters before, a decrease of dissipated energy that through heat that we also see in the slides before, and in general less frost production. So actually, also in this case, we are proving, this is the last slide for Vita, and then we go for good. We saw that for Vita actually is decreasing really reactive oxygen species production in the chloroplast, and this is by protecting the photosystems through remodulation, remodeling the membrane of the chloroplast, as well as putting in place different modulation of the photosystems themselves. And the plants don't do anything. Detoxification has its cost. So if the plant is reducing the cost, the energy used for detoxification, it has more energy to grow. And that's why we see differences in the biomass and in the yield. For good, actually, like Zai said, that this is both for good and for Vita. That's true. It's true. Also, for good has, although it has been developed for another purpose, to increase nutrient use efficiency, and it's foliar applied, and again, it can be applied in all crops in comfort and stress conditions. It has some, how can I say, some mechanisms, some mode of action that is similar to for Vita, some that are similar to for Terra. Because actually we used them in the same experiment. I just didn't want to show all the data. And so we could see a decreasing in ROS with everything that comes after that. And we see an improved also in this case, there is an increase in the levels of the orthophosphate transporter of the chloroplast, PHT 2.1. Yes, 2.1. For good, it's applied in a slightly different way compared to for Vita, because it's also in this case, it's applied like for Terra, but in foliar application, okay? Although since it has effect also on reactive oxygen species, yeah, it can be applied also on other types, but we suggest to apply it where for Vita should be applied. I will go on just with this few more slides, and then we can, if you have questions, you can ask. Just to say that the Plants for Plants journey doesn't stop here. Also, Marine said that in the email. Yeah, because to arrive to these 3 products, the journey was quite long indeed. We had a lot of trials. We had demo trials, thanks all to you. We had European-funded projects like the LIFE, and thanks to that we are here together. And we had a lot of collaborations with you, with other companies, with universities. We did a very good job, I think. We studied the mode of action. So, okay, it works, but why? Why we are not completely satisfied with that. We still want to know much more about our products. We want to know more about how they work. If we can apply it to many different other crops in other stages, or if they can have an effect also on other type of stresses, because we just focused on drought and comfort. Yes, comfort, I put it like that because actually, as I said, oxidative stress is always the plants are never in comfort, completely in comfort. I don't think also in the Dutch greenhouses they are completely in comfort, I think. So we want to know much more, and that's why we will continue to do trials and to provide much more information also to you and to the farmers. But the pipeline is starting to fill with other products. This is one of them. I don't know if someone already heard of that. Zahi yesterday briefly told something about IL-17. This is still in development because it's pretty advanced, but still not as advanced as the ones that are in the market. And in this case, this biostimulant, I wrote here where biostimulation meet biocontrol, because we started to study it to improve the plant's strength against biotic stress. And we proved actually with transcriptomic analysis that it's actually what is happening. We don't have a direct effect on the fungi, although we see some effects on Petri, but we don't think we have a direct effect on the fungi. It's more through the fortification of the plant. Just to see very quickly one picture. So this is a trial we performed on grape where we reduced the amount of fungicide by 30%, fungicide against Plasmopara viticola by 30%. You see that we got, with additional IL-17, we got higher yield. Okay, it's not statistically significant, but still it's nice. And this is what you can see visually. This was a very particular trial with a really strong pressure of Plasmopara. But you see, I think you can see here, the bunches treated with the minus 30% copper, and that's it. And bunches treated with the minus 30% copper plus IL-17. So it's very promising, and we hope to develop it more in the near future. But these pictures show that there are, you see, there are a lot of other stresses that are hitting the plants, not just water stress, all of them are causing oxidative stress and everything. But we think that since plants have more fantasy, and you saw exactly the slide that Adriano showed yesterday with a lot of all those molecules that the plants can have, we can find in nature a lot of help, a lot of answers to help our crops thriving in this changing environment and to produce more for us. So if you have any questions, I'm here to answer. I'm here, Laurent is here, Adriano is here, Zahi is here to answer. I'm afraid of you.
Oh, let's talk.
Maybe if allow me just to add something on this slide.
Yeah, please.
Yeah, so just if you see on this slide, it's pretty much the conclusion of what Christina said, because not as scientifically deep, but this is what is happening. When we start from the clone or from the seed, we are at 100% genetic heat potential. You buy the seeds, you know that these seeds in some specific conditions will give you X ton per hectare, let's say. But then what happens, and what Christina explained really deeply about the oxidative phenomena, we call it, is that we, just by breathing and just by growing, the plant will lose what you have from the 100% genetic heat potential through the loss to natural oxidation. So technically speaking, we all start at this 100% and not on the promise on the seed pack or on the clone that we buy. That we always have some loss due to oxidative phenomenon. And what so far we have been able to achieve with traditional biostimulants is to bring back the potential to this new 100%. And with P4P, what Christina just presented, and thanks to the throughout research that we did with LandLab, is that now we are able to tap on the loss that was previously unreachable, which is, you can see this square that Christina is showing. Normally, you cannot go there because of the oxidative stress that you constantly have on the crops. So today, thanks to the P4P technology, I would say, and to the concept of P4P, that protects us from the oxidative phenomena, we have the chance to raise further higher than the traditional biostimulants. So, commonly speaking, I think it's nice to see that today this is not the limit anymore. We push again the limit from the traditional biostimulants.
Yeah, thank you, sir.
ما فهمتش اشرح لنا يا رب.
هذا
تعمل مخ كثير.
Huh? I lost the last word, so.
لا اشرح لنا الجميع.
sorry, I'll do it also in Arabic.
Yes, please.
ال 100% الجيناتيك يلد بوتنشال بالعربي. ال 100% هذا بس تشتري انت بذر او نوع جار مثمر او الى اخره، عندك بيكون على الكتالوج حاطين لك انه هيدا بيجيب هالقد انتاج بظروف معينه. اللي بيصير بالتراديشنال بايوستيمولنتس اللي بنعرفهم السي ويد والى اخره هول، انت بتعوض الخساره عن النيو 100% الجديد يلي انت خسرته من وراء الفينومينا اللي شرحته هلا كريستينا، انه تاكسد الحاصل بطريقه دايمه من خلال بس انه النبات عم بيعيش ويتنفس ويتعرض لحيالله شيء بالبيئه تبعه عم بيصير فيها الخساره. وبالتالي انت بهالعمليه هون اللي عم بيصير معك انك لانه بي 4 بي بيشتغل على موضوع التاكسد الطبيعي الحاصل بطريقه دايمه بالنبات، ما في ولا بايوستيمولنت بالسوق بيقدر يسيب هيدا الخانه. فبالتالي انت علات ال 100% تبعك اكثر من يلي انت اصلا حصل عليه اليوم بالطرق التقليديه، وهذا كان جوابا على سؤالك امبارح انه انت شو الجديد؟ شو اللي بفرق انك اليوم عم بتصيب خانه لا السي ويد ولا الهيوميك ولا الامينو اسيدز ولا الاشياء اللي بنعرفها اصلا قادر يوصل عليها غير ملعب. وبالتالي اهميه البرودكت والتفكير فيها بعد.
شكرا زين. And that's
He saved you from a lot of questions.
thank you, Zain. Okay, some other questions? Maybe we can have them also later if you want to think much more.
That's why it's so important. You mentioned that the research is going on. Two examples, very recent. Recently, we discovered the effect on soil, on microbiome as well for Terra.
Escados, for instance, like he said with the
And
forbidden.
also regarding enzymes, this is also something
Yes,
that
yes.
is very interesting for us because we will be able to explain more how it works. And now another thing to do and to discover is what about the effect on scan.
I think that was very, very interesting.
We have no idea, but everywhere in the world we observe the same phenomenon, the same effect.
At least 3-4 trials running
Ireland, Turkey, and Belgium.
different
Pretty
locations.
much connected, you know.
Very close to each other, yes. Okay.
I have a question, but later on.
Okay. Later.
Okay, I do apologize because I didn't introduce Christina. Sorry, we wait for the guests, and I hope you will forgive me. Come to Serbia to have fun.
Yeah.
Okay. Then thank you very much for the nice presentation. No, I think it's okay. The next part of presentation will be where is Paul?
Bonus.
Yes. With Paul and Marine. Yes, we will speak now more about how we can sell all of that.
That's the most